Hello, my name is George Kosmidis. I'm a senior scientist in Ncardia, and today I will be presenting a short overview of our poster presentation for the 2020 virtual ISSCR conference. Cell and gene therapies show promise in providing new therapeutic strategies for a wide range of indications. One of the major bottlenecks for cell therapy is robust and large-scale cell manufacturing. Technologies based on human-induced pluripotent stem cells hold great promise to accommodate the future demand for cell therapy. To enable robust and scalable production of iPS-derived cells, we have developed a platform comprising of state-of-the-art manufacturing systems, which allows us to optimize processes at a small scale in order to validate the most promising conditions at mid-scale and to bring the process to the necessary billion scale. These systems allow full automation and conduction in closed systems, resulting in cultures with comparable characteristics from batch to batch. Closed system parallel processing with increased automation is also critical to minimize error and contamination from human interaction with cell products. As an example, using a quality by design approach, we established a controlled stir tank bioreactor process that is shown to routinely yield high numbers of iPS-derived cardiomyocytes. We demonstrate a robust and controlled process for large-scale manufacturing of iPS-derived cardiomyocytes to a purity of 95% in a serum-free protocol. The resulting process provides a relatively mature cardiomyocyte model, recapitulating a human cardiomyocyte's contractile and electrophysiological profile with high predictivity in cardiac toxicity and efficacy assays. Bioreactor iPS cardiomyocytes display the standard morphology and the typical electrophysiological profile as observed with any other iPS cardiomyocytes. As seen in our next panel, the pharmacological responses of bioreactor iPS-derived cells to hERG channel, calcium channel, sodium channel blockers, and adrenergic stimulation are as expected. A blinded study with bioreactor iPS cardiomyocytes and a number of CIPA reference compounds confirms a ranking in terms of their severity towards the electrophysiological phenotype, further confirming their excellent physiological nature. Ncardia now possesses the process development tools that enable us to develop manufacturing methods for iPS derivative cells of various tissues, from small scale to medium and large scale output using our platform. For any questions relating to this poster, do not hesitate to contact us or visit our website at www.ncardia.com.